Imagine, for a second, that you could actually look inside an animal's brain and see the exact cells light up when it gets a reward. I mean, see it in real time. Or watch the specific circuits that fire when it learns something totally new. Well, today, we're going to dive into an incredible technology that gives scientists that exact power. It's called fiber photometry.
So let's just jump right in, because this is one of the biggest questions in all of neuroscience, right? How do you catch that spark? You know, that fleeting moment that connects a specific behavior, like a mouse finally figuring out a maze, and that split-second burst of electricity deep inside its brain.
And believe me, for decades, this wasn't just a question, it was a massive wall. The brain's signals were just too fast, too complex, and buried too deep to connect them to what an animal was doing moment to moment. But then, a new generation of tools started to chip away at that wall, and they gave us something so elegant, it's like installing a magic window directly into the living brain.
And that window is fiber photometry. And honestly, this is the best way to think about it. It's like scientists can now go into the brain, find a very specific group of neurons, and just hand them these tiny biological glow sticks. And the instructions are simple. Hey, whenever you're active, I just need you to glow for me. And just like that, invisible electrical chatter becomes visible light that we can actually see and measure. It's just fantastic.
Okay, so here's our game plan for this explainer. First, we'll talk about the brain's secret language. Then, how we finally gave neurons a voice. We'll peek inside the scientist's toolkit to see how it works, figure out how to turn a signal into actual meaning, and finally, we'll talk about why it's such a powerful but still imperfect tool.
So, at its core, the challenge has always been about listening in. I mean, think about it. When an animal does anything, thousands, maybe millions of cells are firing all over its brain. So how in the world can we isolate the one conversation happening in one specific area at the exact moment that really matters?
So how did we even get here? I mean, a technology this cool doesn't just appear overnight. It's really built on the shoulders of decades of innovation. So let's take a quick look at how we learned to listen with light. The whole journey really kicked off back in the 80s with these early ideas about measuring calcium in cells to see if they were active. Then, step by step, the tech got better and better. But the real revolution, the thing that changed everything, was the development of modern genetic sensors.
And this is where it becomes a total game changer. We can now target only the specific types of neurons we care about. So say you only want to listen to the dopamine neurons, the ones that run the brain's reward system. With these new sensors, you can give that glow stick just to those cells and completely ignore all the noise from their neighbors. It's a gigantic leap forward.
Of course, for all this to work, you've got a whole system of hardware working together behind the scenes. So let's pull back the curtain and take a look inside the neuroscientist toolkit. You know, the setup is actually brilliantly simple in its design. At its heart, you've got three main parts: a light source, which is usually just an LED, a fiber optic cable that is literally thinner than a human hair, and then some filters and a really sensitive detector to pick up the signal. And the fiber optic cable, this is the really clever part. It acts like a two-way highway for light. It sends light in to get the sensors ready to glow, and then, when the neurons actually fire, that very same cable carries their faint little glow all the way back out of the brain to the detector. That little bit of returning light, that's our data. Okay, so we've got the theory, but what does a real experiment look like in the lab? Well, it starts with a very precise surgery, where a harmless virus carrying the gene for our glow stick gets injected into the brain. Then, that tiny little optic fiber is implanted right above that same spot. But you're not done yet. In fact, one of the most important parts is just about to start: the waiting game. That's right. Scientists have to wait 2, sometimes even 3 weeks for that virus to do its job and get the neurons to actually start making the glowing protein. It's a great reminder that patience is absolutely a virtue in this line of work. All right, so weeks have passed, the experiment is up and running, and we're getting data. But what we have are just these squiggly lines of light on a computer screen. So how do scientists turn that into a real story about the brain? Well, the very first and most important step in that whole process is a little calculation called delta F over F. Now, I know, it might look a little intimidating, but it is the absolute key that unlocks all the meaning hidden in those flickers of light. What it does is actually really simple. It just normalizes the raw signal. So instead of telling you the absolute brightness, it tells you the change in brightness from its own baseline. Think about it like a dimmer switch on a light. Instead of saying the bulb is at 200 lumens, you just say, hey, the light just got 50% brighter. This is what lets us confidently compare recordings across different days or even between different animals. And this is what leads to that big aha moment.
Once the data is processed, you can see it clear as day. A huge spike in dopamine activity the exact moment an animal gets a little sugar reward. This is it. This is that concrete, instantaneous link between brain activity and behavior that we've been looking for.
But the real magic, the really cool part, happens when we sync this brain signal, frame by frame, with a video of the animal. Now we can ask, what was the animal doing the instant that signal peaked? Oh, it was reaching for a lever. What was it doing when the signal dipped? It was sitting still. This pairing is how we truly start to translate the brain's complex language.
Now, there's no doubt that fiber photometry is revolutionary. But like any powerful tool, it's got its strengths and it's got its weaknesses. So let's just lay them all out on the table.
The advantages are like superpowers for a neuroscientist. We can finally listen to the deepest parts of the brain where microscopes could never go, and we can do it while an animal is just freely moving around, behaving naturally. And we can do this for months at a time. But every superhero has their kryptonite. The big weakness here is spatial resolution. We're not seeing individual cells. The signal can also get messy if the animal moves a lot. And finally, remember, we're measuring calcium, which is just an indirect clue. It's like seeing smoke, not the fire itself.
So this is a really important point to remember. The signal we're recording isn't the quiet whisper of a single neuron. It's more like the average cheer from a crowd of hundreds of neurons all firing together. It's a population signal. So when we interpret these amazing results, we always have to keep that limitation in mind.
So at the end of the day, fiber photometry has given us this incredible new way to listen in on the brain's symphony as it happens live. But what's really exciting is thinking about what comes next. With new and better sensors and systems that will let us listen to multiple brain regions at the same time, what great secrets are we about to unlock? How memories are formed, how we make decisions, maybe even consciousness itself. We have never been closer to cracking the brain's code, and it looks like the future is going to be very, very bright.